Hey guys, today's lesson is really focusing on part 4 of our Meiosis genetics heredity unit. We're going to look at three major things on here: incomplete dominance, codominance, and multiple allele situations. So really just some special cases where our typical monohybrid crosses don't always do these situations justice. So to begin with, we're going to look at incomplete dominance. Incomplete dominance, the definition of this, or the one we're going to work with, is that neither allele is completely expressed, and it creates a new phenotype that's really a blend of other traits. So our example that's over here on the right that you can see is that a red flower is then bred with a white flower, and the resulting cross is a pink flower. So in this case, we have three possible phenotypes, or what we see. We could see red, we could see white, and we could see pink. So here's our 1, 2, and 3. In this case, the red one and the white one blend together, and they make the pink. So in this case, what we're looking at is R and W is the alleles. The C is just a way that we write this to show that the codominance is happening. So we can bring the R down, and then the C with the W over. You can see that the same thing happens as we go through. And we end up with a Punnett square. That's really just the same as doing a monohybrid, only they have these little superscripts on top, basically. But what we see to tell us what color this is, R and W together, a red and a white allele together, there's no longer a dominant gene allele that covers this up. So we see that the red and white alleles together create this new phenotype of pink. The easiest way to remember is when we see capital letters here, there's no lowercase. There's no true recessive allele, which means one is not ever going to be covered up by the dominant allele. They're both, in essence, dominant alleles. Our next situation looks pretty much exactly the same, but instead of getting a blend, we get a brand new phenotype. We get something totally new. So it's not really a mixture anymore. We get both kind of being expressed together. And I think that's the key takeaway here. So while our Punnett square is going to look identical to that other one, it's going to mean something different now. So instead of using flowers, use cattle. We could use the flower, but I don't want you to get them confused. Cattle here can show this really well. So we get this new phenotype, right? This new phenotype is going to be codominant. Co is happening together, so these are going to happen together. When we cooperate, we work together. In codominance, both of these alleles are going to work together. So what happens is we get these red cattle, and we cross them with white cattle, and the resulting situation here is what we would define as a roan cattle. And a roan cattle, for those of you that don't know, looks like this right here. So it's got the brown and the white expressed in different amounts. And that's our new phenotype here. We have a brown and white kind of spotted cattle. Same thing, exactly the same thing as we did in the last cross, only it's going to mean something different. All right. Now, if we did this with parents that weren't identical genotypes, we would see that we could still end up with some brown and white cattle separately. So if we did an F2 cross and we took these two guys right here and bred them together, we would see that some would end up being brown and some would end up being white. So we'll practice some of those codominance problems in class a little more specific. The key is that both are expressed. Finally, we're going to look at multiple alleles. Multiple allele situations are when there are more than two alleles that exist. So before we talked more about in codominance and incomplete dominance, the idea that phenotypes are expressed differently. Now we're looking at what happens when there's just three different alleles altogether instead of two like we normally do. So with blood types, this expresses pretty well. Looking at blood types, we have different A, B, and O combinations that we can do. So we really have three here. And this really talks about the difference in how that all works out. It has to do with antigens, and that's probably more complex than we really need to understand. But just know that there are different blood types possible from combinations of those three. So how we write this, and I wanted to kind of show you the breakdown too, just in case you don't know your blood type. We could take a look and see kind of where you fit in with terms of population. Type A blood alleles could be this in our square or this, all right? So you'll see again we have I as the base for this, and we have a superscript with A's and B's. If there's nothing on it, then that's our O allele. All right, we don't write the O up here, but we just know that that's what it's referring to. B, again, could be both B's, or it could be one B and then an O here, right? So we just leave it blank. Type AB alleles have to have the A and the B together. That's the only way to do it. And to be type O, you have to have no A's or B's present at all. So think of that as the ultimate recessive allele. A blood type cross, we'll go ahead and do this. And we can use blood types to tell us all kinds of information. In this case, possible offspring of this individual is going to be what blood type here? We have type A. There's an A allele and nothing on this one, so that's an O. So this A is expressed. And same here, this individual is going to be our type B. So type B and a type A blood type individuals have children. Then we're going to break this down in the Punnett square and see what they end up coming up with here. So we have our I A and I B. And then here we have, bring this I, capital I over with the B allele, then the small I down. Remember that's the O, but we don't need to write it. Same thing here, bring this big A, I, the A allele, and the small I over. And this one is a small I, down and over. So we have, in reality, genotypes of the offspring. We have 3 different genotypes present, or blood types that they could be. The first one is right here. That's going to be an AB blood type. All right. The next one is going to be a B, type B individual. The next one is going to be a type A individual. And then the fourth one here is going to be a type O. So a parent who is A, heterozygous for type A and heterozygous for type B, could produce a child with all of these different blood types, each one with a 25% chance here. So phenotype is what they actually are. In this case, the phenotypes are really easy to write down. It just looks like that. There's AB. Next phenotype is type B. Next phenotype is type A. Next phenotype is type O. Right? In percents, 25% chance for each one.